In this video, we're going to be looking at how active transport processes that use ATP helps to move stuff from one side of the membrane to the other. And so we'll start by looking at the idea of active transport. With active transport, we can move materials against the concentration gradient, move them from where they are in low concentration to where they are in high concentration. So from low to high this time. So here you can see we've got, this is the nutrient that we're trying to get, but here we see lots of nutrient on this side that is filling up the interior of the cell. But because we are moving against the concentration gradient, energy is required to make this happen. And energy is supplied in the form of adenosine triphosphate, in the form of ATP. And similar to facilitated diffusion, a carrier protein is used. But this time, rather than just having the nutrient cause the change in the shape or the conformation of the carrier protein, it's ATP energy that changes the shape. So when the nutrient lodges here in the carrier protein, then ATP energy is delivered, and in order to harvest that energy, we break off one of the phosphate groups. So that's what it's showing here, that there's some inorganic phosphate broken off, and we're left with an ADP molecule, adenosine diphosphate molecule. And that ADP molecule and the phosphate group later can be put back together again to recharge the battery, if you will. But ATP releases its energy, the carrier protein changes shape, and boom, the nutrient winds up inside the cell and moved against its concentration gradient. So that is active transport when we talk about it this way. But active transport isn't the only way that ATP energy is used to move materials from one side of the membrane to another. We can also move materials in what is referred to as a group translocation reaction. And rather than using ATP to do this, we require a different energy source. It's called PEP, or phosphoenol pyruvate. And it's another high energy molecule, sort of similar to ATP in that respect, that it carries lots of energy in its chemical bonds. So doing group translocation reactions also moves materials against a concentration gradient, and it uses PEP as the energy source to do this. So what happens with a group translocation reaction is that something like glucose is brought into the cell using this PEP energy, and there are several other steps. We're not going to worry too much about what those other steps are requiring, but through a series of steps that involve several other proteins as well as some phosphate groups associated with those proteins, then we're able to move glucose into the cell and at the same time change glucose so that it's no longer able to move back out again. So we turn it into glucose 6-phosphate, where we add a phosphate group to the glucose molecule, and now it's a material that is different actually than glucose. It's a new molecule that cannot be transported back outside the cell. And then the glucose is later on used as we pull off that phosphate group. Then it's converted back to glucose and then can be used to make ATP. And glucose isn't the only thing that's moved into cells by these group translocation reactions, but it's a great example of it. So with group translocation reactions, we bring material into the cell, modify it chemically so that it can't be moved back out of the cell, and then the material can be used later on. And it's done with an energy source, this time PEP, or phosphoenol pyruvate. PEP, or PEP, is the only thing I'm going to ask you to learn or know about that. You don't need to spend a lot of time trying to memorize phosphoenol pyruvate at all. So Bacteria use the group translocation reaction to bring glucose into their cells, and we see similar processes to this in eukaryotic cells as well. So a second way to move against the concentration gradient, active transport is one with ATP, group translocation reactions with PEP are another. So there are still ways that we can bring materials into the cell that require energy, and a third of these is endocytosis. So endocytosis occurs when part of the cell membrane makes this little inpouching here, and that draws in a material into that inpouching. And over time, that little pouched area gets narrow and narrow until eventually it gets pinched off, and now we've got whatever the material is. And they're showing that a bacteria has been brought into a eukaryotic cell this way. And so we bring that material into the cell in the form of a vesicle. And then the next thing that happens with endocytosis is that we generally go and digest whatever was brought in. And so we use lysosomes. Those who remember the lysosomes have in them enzymes that can digest materials. And then we produce this vesicle that is filled with digestive materials. And then the good stuff, the things that we want, can be transported out. And then we're left then with this residual body, this little bit of stuff that is waste material, essentially. So that's endocytosis, bringing materials into a cell. We can get a little fancier with endocytosis to bring in not just everything that's outside the cell, but very specific materials by doing something called receptor mediated endocytosis. So this time, rather than just picking up whatever happens to be outside the cell, we can stick little receptors out at the surface of the cell that are very attractive to a particular material that we want to bring in. So we'll not worry about what that stuff is. I stole this from from some other textbook that I've got. And so if we want to bring these little molecules here, we could think of them as glucose, and this is how eukaryotic cells bring glucose into their cells, is they stick out receptors that will bind the glucose there at the surface of the cell. And once all of the receptors fill in an area, then we do endocytosis. So we see this inpouching happen until eventually we get a vesicle, begins to form that has all of those receptors that are all filled up inside of it. Until eventually then we get this vesicle that has everything inside of it. Then, once again, we can bump a little digestive lysosome up against it and digest off the material that we wanted to bring inside the cell, and then move that material out into the cell. And now we're left with the vesicle and a bunch of empty receptors on it. And if the cell wants to keep bringing whatever that material is into the cell, then it can go and plop that vesicle with its receptors on the cell back at the cell surface again. And when you bump a vesicle up against the cell surface, then the membranes will fuse right there where they touch to each other. And that will then, if I can, let's see. So now the fused membranes will open up, and we've got the vesicle here with its receptors on it. And eventually, as this wrinkle in the cell surface smooths out, then the receptors are back at the surface, ready to pick up more of the goods again. And as soon as they fill up, then we do endocytosis once more, digest off the good stuff, and absorb the good stuff into the cell, and then return the receptors back to the cell surface again. So that's receptor-mediated endocytosis. And any time we're doing endocytosis, it requires cellular energy to do so. So ATP is going to be required for this process, as well as regular endocytosis as well. So receptor-mediated Endocytosis is just endocytosis with a receptor that can be very specific about what we're bringing into the cell. And then last of all, the last bits that have to do with using energy to move materials across the cell membrane is, I'm sorry, we're not quite at the last bits. If we're back to explaining then two different types of endocytosis. If endocytosis brings a solid material into the cell, then that is referred to as phagocytosis. Phagi, that P-H-A-G prefix, is about eating. So this is cell eating. And if what the material is that is being brought in is fluid, then we refer to that as pinocytosis. So phagocytosis brings in food chunks. Pinocytosis brings in liquid materials. And almost all cells in our body do pinocytosis all the time. They're busy bringing fluids into the cell, replenishing fluids that way. And then, if we look at the far end of phagocytosis, once phagocytosis is finished and we have digested the materials, then that undigested residual body here, just like we saw with receptor mediated endocytosis, where we ended up putting the membrane back on, putting those receptors back onto the membrane, we can do the same thing with this residual body. We bump it, we bump it up against the cell membrane. Now, as the cell membrane fuses with the vesicle, then that opens up and dumps everything out via a process called exocytosis, also requiring ATP energy to do that. So phagocytosis brings in chunks, pinocytosis brings in fluid, and then exocytosis returns the membrane back to that membrane that we turned into a vesicle back to the cell membrane, and it dumps materials outside the cell. So here we see exocytosis then kind of mentioned on its own. Again, a vesicle that's filled either with cell product or with waste materials. So we definitely get rid of wastes after phagocytosis, but cells will also dump out cell products that they make. So, for instance, an example of this might be a cell that is producing sweat, and by exocytosis it dumps out sweat product, or the mammary gland cells that are in breasts. So those mammary gland cells, they dump their proteins and fats and sugars out into the ducts of the mammary glands by exocytosis. So as the vesicle fuses with the cell membrane, then materials are released because they are now outside the cell. Okay, so there we are. That's all the active transport ATP requiring processes. Active transport group translocation reactions, endocytosis, including phagocytosis and pinocytosis, and then exocytosis that dumps everything out. Oh yes, and receptor-mediated endocytosis that brings very specific materials because we bind them on receptors at the cell surface. You know what to do if you have questions. Talk to you.